Hello, welcome to Module 2, study session 1, which is basically going to conclude fatty acid oxidation. Okay. Um, we won't waste much time here because we've already covered fatty acid oxidation in study session 4 of Module 1. This is the remaining parts we've not covered. Basically, if you remember, we talked about beta oxidation whenever we mentioned oxidation of fatty acids. And if you remember very clearly again, we focused on the oxidation of even numbered fatty acids, 12, 14. Basically, I think we more or less talked about the oxidation of palmitate, which is a 16 carbon fatty acid. 16 is an even number, right? Now, what if you are dealing with oxidation of an odd numbered fatty acid, maybe 13 or 15 carbon chain fatty acid, for example? How do you deal with that, right? So we're going to cover that here. Okay, so this is just an introduction uh, according to your manual, right? Your note course material. Although beta oxidation is a major pathway of fatty acid oxidation, it is limited to the oxidation of even-numbered saturated fatty acids. Just like I mentioned earlier, even-numbered saturated palmitate is even-numbered 16 carbon chains without any double or triple bonds. So it's saturated. It has only single bonds, right? Okay, so other fatty acids, unsaturated fatty acids, for example, in biochemistry, we have the unsaturated form of palmitate is palmitoleic fatty acid, for example. Palmitoleic is unsaturated. It has a double bond. It's even, but it has a double bond, for example. So what if you have an unsaturated fatty acid? How do you oxidize it? Through beta oxidation. It will mean we would have to have additional steps to eliminate the double bond, right? So those with odd number of carbon atoms also have to be taken. You have to take probably a different approach before going through the conventional beta oxidation. And if beta oxidation is not obtainable for them, then you have to go through a different type of oxidation to actually metabolize uh, odd number, odd numbered carbon length fatty acids. Okay. So also animal tissues contain minor pathways that involve oxidation of fatty acids as the alpha and omega carbons. Okay, so if you remember, I think it got to be in this slide. I think we've mentioned alpha and omega carbon atoms. If it's not here, then I probably forgot to show that here. Okay, I'm coming. Please give me a minute. Um, yes, it's okay. So in biochemistry, whenever you hear the alpha carbon, beta carbon, if you remember chemistry very well, each compound has a functional group. This, we're talking about fatty acid. So of course you have an acid functional group, carboxylic acid, right? Now this makes this carbon one. This is the functional group. The carbon atom, this that follows the functional group is your alpha carbon. The carbon atom that follows the alpha carbon is a beta carbon. The carbon atom that follows the beta carbon is a gamma carbon. So beta oxidation occurs here. It, it cleaves, the cleavage happens, and you have two carbon atoms, two carbons of acetyl-CoA going out because of beta oxidation, right? Gamma carbon, uh, delta carbon, epsilon carbon, up to the last carbon on this side, which is the omega carbon. So we have, apart from beta oxidation, we have alpha oxidation, as well as omega oxidation of fatty acid. Now, for normal saturated, even numbered fatty acid, beta oxidation occurs. For certain types of fatty acids, we go for alpha oxidation. The body goes for alpha oxidation. While for certain types of fatty acid, the body goes for omega oxidation, which we are going to discuss, right? So I think it makes sense now. So basically, this is what we are talking about here. The products of alpha and omega oxidation can enter beta oxidation. Of course, ketone bodies, for example, which we are going to discuss, serve as alternate source of fuel during periods of fasting or starvation. The brain, for example, uses glucose exclusively, but in the absence of glucose, like in diabetic condition, the brain can use ketone bodies. Disorders of fatty acid oxidation include carnitine deficiency. You remember carnitine, right? Which transports long-chain fatty acids from the outer mitochondrial membrane into the inner mitochondrial membrane. So disorders of fatty acid oxidation can include the deficiency, which prevents the transport of long-chain fatty acids, and the body or the cell becomes unable to oxidize long-chain fatty acid because of carnitine deficiency. The Refsum's disease, Jamaican vomiting sickness, Zellweger's disease, and ketoacidosis, which of course we're going to discuss in a minute. All right. Other mechanisms of fatty acid oxidation, which I think we've already discussed. This is just a preamble. Okay. So, while double bond that is generated between the alpha and beta carbons is the first step of beta oxidation. And the trans is in the trans configuration, double bonding. Okay, I see. Let me make this thing easy. I've already explained the beta, alpha, and gamma carbons. To just simplify this, okay, what will happen is I just take the time to explain, and then you read on your own. If you don't understand anything, you can ask me questions in the forum. Okay. So basically, beta oxidation affect unsaturated fat only. No, of course, false, because saturated fats are, of course, affected also by beta oxidation, like we treated last week, right? So now, how do you oxidize fatty acid containing an odd number of carbons? This is easy. We treated fatty acid containing even number of carbons, 16 carbons. So by the time you cleave 2 carbons, because that's what beta oxidation do, the beta oxidation will give you 2 carbon atoms, right? 1, 2, 1, 2, 1, 2. So by the time you reach 16, you're still going to have 2 carbon atoms because it's even. But if it's odd, by the time you reach the end, you end up with 3 carbons, right? Not 2. So, and those 3 carbon atoms now become what we call propyl-CoE. Instead of acetyl-CoE, now you have propyl-CoE. Now this propyl-CoE is metabolized, and the metabolic pathway is shown here. Let me show this as a slideshow. Oxidation of propyl-CoE produced by beta oxidation of odd number fatty acids. The sequence involves the carboxylation of propyl-CoE to D-methyl-malonyl-CoE and the conversion of the latter to succinyl-CoE. You remember succinyl-CoE from your TCA cycle? If you remember, please, if you don't remember, please refer back to our lecture on TCA cycle. Succinyl-CoE is produced from succinate, right? Succinyl-CoE is converted to, I think, fumarates, okay? Fumarate is converted to malate. Malate is converted back to oxaloacetate in the TCA cycle. All right. So this conversion requires epimerization of D to L-methylmalonyl-CoA. This is a reaction. I don't know if you still remember, if you've been taught carbohydrate metabolism or just biochemistry of carbohydrates, you are going to encounter this word epimerization. D to L-methylmalonyl-CoA, followed by the remarkable reaction in which substituent of, all right, let's just make it easy. So if you have a 15 chain fatty acid, for example, a fatty acid with 15 carbon atoms, so beta oxidation will cleave 2 carbon atoms successfully until you reach the last 3, because it's 15. It's not going to be even. The last 3 is propanyl-CoA. Propanyl-CoA is carboxylated by propanyl-CoA carboxylase, right? In the presence of ATP and in the presence of bicarbonates, right? Which produces ADP plus Pi, and then the CO2 component here is added, right? You see this, you don't have this component here. Well, here, because of this carboxylation, due to the addition of this bicarbonate, you have this, right? And this is now D-methylmalonyl-CoA. Methylmalonyl-CoA is epimerized. That's the name of the reaction. The enzyme that does this is an epimerase from D to L, right? L-methylmalonyl-CoA. Now, L-methylmalonyl-CoA can go through a forward or reverse reaction. The enzyme that does this is a methylmalonyl-CoA mutase in the presence of coenzyme B12, cobalamin, right? Cyanocobalamin, which are vitamin B12. Presence of this coenzyme. That's why vitamins are important, you know? They play roles as coenzymes for some reactions in your body. Now, this succinyl-CoA can go into the TCA cycle, or succinyl-CoA can be converted back to glucose if your body requires glucose. The reaction, the pathway through that converts succinyl-CoA to glucose, I think is the glyxylate pathway. You can confirm that, please. But I think so. Through the glyxylate pathway, then gluconeogenesis converts succinyl-CoA to glucose. Okay. This is quite interesting. So basically, this explains everything here. Everything you're reading here, that's the pathway. I think it explains everything. So sometimes as biochemists, we prefer to just look at the pathway instead of reading all this story here. Of course, the story is part of everything, but if you understand the pathway, it simplifies everything, okay? Now, alpha and omega oxidation of fatty acids, right? This is it. Everything is here. But I'll allow you to go and read this on your own. I'll just show you what I have here, which simplifies everything for me, to be honest. Okay? So alpha oxidation. What's alpha oxidation? Okay? So alpha oxidation is defined as the oxidation of fatty acid that has methyl group at the beta carbon. Usually, your fatty acid I apologize. I'm trying to get to. Usually, your fatty acid looks like this. This is your alpha carbon. This is the functional group, right? The carboxylic acid, but here it has been modified by addition of CoAs, which gives you the component of acetyl-CoA after beta oxidation occurs, which will cleave this, right? I remove this to get your acetyl-CoA. Now, if you look at this CH2, right? You have 2 hydrogens attached to this carbon, 2 hydrogens attached to this carbon. You don't have a methyl group. A methyl group is a CH3, and you don't have a CH3 here. Now, this is a normal fatty acid, okay? Now, in cases where you end up with fatty acids that look like this, right? This is the functional group, the carboxylic acid. Okay. This is the alpha carbon. This is the beta carbon. At the beta carbon, we are supposed to have CH2. CH2. Now, this here is a carbon. This is the beta gamma. This is the delta carbon. The delta carbon has a CH2. The reason why you don't have this is because it's hydrogen. We don't need to draw this. But when you see this in chemistry, this is a methyl group. And that's why they had to draw it to delineate methyl group from, to, how do you call it, to differentiate a methyl group from a normal hydrogen, which does not have this bond here, right? So this is a way of representing chemical structures. In order to be good in biochemistry, you have to be average at least, or good in chemistry, okay? Um, so this is the carboxylic carbon, the alpha carbon, the beta carbon has a methyl group. So this isn't your normal fatty acid you're used to, right? And that is why we have to carry out what we call alpha oxidation instead of your conventional beta oxidation. Now, the methyl group at the beta carbon, with the removal of one carbon unit adjacent to the alpha carbon from the carboxylic acid end to form a CO2. So it is the oxidation of fatty acid that has a methyl group at the beta carbon with the removal of one carbon unit adjacent to the alpha carbon, which I'm going to show you through the reaction. Alpha oxidation occurs in those fatty acids that have a methyl group. Of course, we've said that at the beta carbon, which blocks beta oxidation, right? Of course, the methyl group is the beta oxidation does not recognize something like this. So it's actually compromises beta oxidation. Now, an example of such substrate, such fatty acid that has a methyl group at the beta carbon is phytanic acid, which I've seen an example in your lecture note, which is present in milk or derived from phytol present in chlorophyll and animal fat. Okay. Peroxisomes is the cellular site for this alpha oxidation, okay? And no production of ATP here. So the steps for alpha oxidation involve the activation of phytanic acid, hydroxylation, that's the addition of a hydroxyl group, removal of formyl CoA, CO2, oxidation of pristanal, and then beta oxidation of non-pristanic acid. After these initial modifications from 1 to 4, you can now conduct beta oxidation. Okay, so here we are. This is your phytanic acid. So phytanol CoA synthetase with also ATP as input produces AMP plus API, and the phytanic acid is now modified to phytanoyl CoA, and phytanoyl CoA is hydroxylated, right, to form 2-hydroxy. A hydroxy group is added, phytanoyl CoA, and you need alpha ketoglutarate, which produces succinate. This is a coupled reaction from the TCA cycle, right? If you remember. All right, alpha ketoglutarate dehydrogenase produces succinate, right? I hope I'm right, okay? So anyway, the presence of this enzyme, we have this, all right? 2-hydroxyphytanol-CoA is broken down, is lysed. Let me use the word lyse to pristanal. And here, a lysis will remove a molecule of formyl-CoA or CO2. And pristanal is now oxidized to pristanic acid in the presence of aldehyde dehydrogenase. And here, NADP is reduced to NADPH. There's always a balance, right? As pristanal is oxidized, something has to be reduced, right? So the coenzyme NADP is reduced to NADPH. So basically, this is, you have your pristanic acid now. And the pristanic acid will now undergo beta oxidation. Of course, it will be activated. And then the beta oxidation proper will occur to produce 2-methyl-propynyl-CoA plus 3-acetyl-CoA plus 3-propynyl-CoA. Okay, so this is structurally now what's going on. I think it makes more sense now if I show it like this, okay? So what really happens here? First, your phytanic acid, right? Which has a methyl group at the beta carbon here, is converted or modified to phytanol CoA in the presence of this enzyme, like we mentioned earlier, right? Now, phytanyl-CoA with the methyl group still at the beta carbon is hydroxylated, right, to alpha hydroxyphytanyl-CoA. The hydroxyl group is added at the alpha carbon, right? The beta carbon still has the methyl group, right? This is your alpha carbon, this is your beta carbon. Now, alpha hydroxyphytanyl-CoA lies will now lies a molecule of CO2, which is formyl-CoA, right? And look what happens here. This goes out and you end up with this. What really happened? That's the question you should ask. Hmm? Your carbon here is still there, right? This carbon is still there. This is gone, right? This is gone. This is still there. And now because we've reduced the number, the chain length, this now becomes your functional group, cristanal. This now, the COH here becomes your functional group, and the methyl group, which was originally on the beta carbon, becomes the alpha carbon. Now the methyl group is on the alpha carbon, not the beta carbon. The presence of methyl group on the beta carbon blocks beta oxidation, but if it's present now at the alpha carbon, the beta oxidation is not blocked anymore, right? Do you understand what happened here? It's quite very clear, okay? This lies this. And what you have now is your COH, which the reaction will now introduce this double bond for a balance chemically, right? Now, aldehyde dehydrogenase will now convert this pristanal, which is an aldehyde, to a carboxylic acid, pristanic acid, where COOH is your functional group. This alpha carbon now has the methyl group, not the beta carbon. And now, since the methyl group has been eliminated from the beta carbon, beta oxidation can now occur. You remember a beta oxidation, right, from what we did earlier. And now, since phytanic acid has an odd number of carbon atoms, you're going to end up with propynyl-CoA, right? And you remember the reaction of propynyl-CoA, just like I showed you. It's going to lead to the production of L-methylmalonyl-CoA, which can be converted to succinyl-CoA, which can be converted to glucose through gluconeogenesis. Okay? All right. So I think I've explained this. Basically, that's it. That's alpha oxidation for you. Now we'll have to discuss omega oxidation. Okay? Yeah, all right. Regulation of fatty acid oxidation. Key ways to regulate fatty acid oxidation. There should be a way for you to regulate everything, right? Without regulation, then things go out of control. The rate of fatty acid oxidation in mitochondria is controlled by regulating the entry of substrate into the organelle. You remember your carnitine, right? The key enzyme carnitine. What do you call it? In the first state, malonyl-CoA, whose formation is the commitment step to fatty acid synthesis, inhibits carnitine transferase. While the enzyme is made very active in the third state. I think it would be made easier if I just use an illustration to explain this instead of reading this. Sometimes your notes can be a little bit confusing. I have an illustration to address this, don't worry. Okay. Acetyl-CoA carboxylase and the fatty acid synthesis, fatty acid breakdown. We're talking about fatty acid oxidation, that's catabolism breakdown. But here they are talking about regulation through synthesis, which we've not discussed, and it's not even part of your curriculum for this course, as far as this course is concerned. Okay, so ketone bodies are water-soluble products of lipid oxidation. We're going to talk about ketone bodies as well. Okay, the extent of this reaction depends on the intramitochondrial We're going to talk about all of these points. Don't worry. I'll just use the pathway here to explain everything. We'll be good to go. All right. Yes. In the clinical aspects. Okay. Now, we've not talked about the omega oxidation. Talk about that and then we'll go to the regulation. I think then we'll call it a day. So omega oxidation, the cellular site for omega oxidation is the endoplasmic reticulum. Yeah, beta oxidation, mitochondria, alpha oxidation, peroxisomes, omega oxidation, cellular site is endoplasmic reticulum, ER. Okay, the omega oxidation occurs at the omega carbon, right? And where is your omega carbon? That's it. Okay, that's your omega carbon. Okay, the carbon most distant from the carboxylic group. This is your carboxylic group, your functional group, carbon 1. Carbon 2 is your alpha carbon. Carbon 3 is your beta carbon, gamma, delta, epsilon. The last carbon far away from the functional group. That's your omega, the lone wolf, but it might not survive because wolves survive best in packs, right? So the substrate for omega oxidation is medium and long chain fatty acids. Okay, and the importance, it is a minor pathway but becomes active when beta oxidation is defective. Yes, the product formed are dicarboxylic acids. So okay, no, sorry. Okay, yes, so this is your omega carbon. Yeah, and this is just we call it summarized pathway to show what happens during omega oxidation. Mixed function oxidase converts this fatty acid, right? CH2 10, 11, 12. So it means here we are dealing with 12, okay, fatty acid with 12 carbon atoms, right? 10 here, plus 1 here, 11, plus 1 here, 12. Okay. Mixed function oxidase in the presence of NADPH. NADPH is oxidized to NADP plus. It means this is going to be reduced, right? And this is the reduction that occurs here on this omega carbon. The omega carbon has to be modified to a carboxylic acid like this before the omega oxidation can occur. Basically, that's what's going on here. Alcohol dehydrogenase will now convert this reduced carbon atom here to a carboxylic acid. This is now reduced while this is now oxidized, right? To this. Aldehyde dehydrogenase. Aldehyde dehydrogenase is converted to aldehyde dehydrogenase, sorry, is now oxidizes your substrate now this to this, of course, just like I said, in a two-step reaction. And the moment you have this, it means beta oxidation can occur. This will now be activated to go ASH and the beta oxidation will occur. And the end, you end up with an adipic acid, right? And succinates. Um, Succinate can go into TCA cycle. It can also be converted to succinyl-CoA and converted back to glucose through the gluconeogenesis pathway. Okay. And now we can talk about the regulation of fatty acid um, oxidation, right? Okay. So if you look at this chart here, yeah, you remember, you remember, you remember trying to remove this. Yeah, sorry. Mm, Okay, so when you take in glucose, for example, dietary carbohydrates, right? Usually after meal, you have a high amount of glucose in your blood. So under normal circumstances, the glycolytic pathway will occur. Glucose will be converted to acetyl-CoA, right? Now, in the presence, fatty acid synthetic pathway. Now, this is what we are talking about, the fatty acid synthetic pathway, which is not part of this study. But if you want to regulate fatty acid oxidation, you have to regulate from both ends through the fatty acid biosynthetic pathway and the fatty acid oxidation. Okay, so acetyl-CoA carboxylase is the enzyme that plays a significant role in the first step of fatty acid synthesis. It converts acetyl-CoA to malonyl-CoA, all right? And this leads to the production of fatty acids through different steps. Now, this malonyl-CoA is known to inhibit carnitine acyltransferase 1. You know what carnitine acyltransferase does, right? Transports fatty acid from the outer membrane of the mitochondria to the inner membrane of the mitochondria. While carnitine acyltransferase 2 to transport the carnitine now that has transported fatty acid already, the carnitine alone will now be brought back to the outer membrane to do the action again. So high levels of malonyl-CoA can inhibit this enzyme. You understand? Okay. Um, this is what this means inhibition. Okay. So this is acetyl-CoA carboxylase. The acetyl-CoA carboxylase can be inactivated by phosphorylation. So phosphorylating this can inactivate it. And if you want to activate it, a phosphatase will remove the phosphorylation, and this will be activated. Let's say in the presence of high blood glucose. When you have high blood glucose in the blood, and some of those glucose will now body will try to convert them to fat, and so that it can be stored in your adipose tissue. So the presence of high blood glucose will dephosphorylate. The enzyme will dephosphorylate or remove the phosphate group that's inhibiting ACC from working. Will be removed using a phosphatase. The phosphatase will remove the phosphate group. Your ACC is now active, and it will now start converting acetyl-CoA to malonyl-CoA and malonyl-CoA to fatty acids. All right? Okay. When your blood glucose is low, right? Your blood glucose is low. It means you need glucose. So you don't want any glucose going into the production of fatty acids. So the presence of hormone glucagon now, which signals that there's a need for glucose, right? Will phosphorylate ACC, acetyl-CoA carboxylate, make it inactive so that fatty acid synthesis will not occur. So basically, these are the steps that happen in the process of regulating fatty acid oxidation. So when there's a need for fatty acid synthesis, we synthesize. When there's no need, we regulate, right? The body does that. So basically, this is what's present in your manual. It's just the best way to explain it to avoid redundancy, right? Now, ketone bodies in humans, and most other mammals, for example, acetyl-CoA formed in the liver during oxidation of fatty acids can either enter the citric acid cycle, of course, right? Acetyl-CoA, we know, enters the citric acid cycle. Sir? Mukhtar Ali, ina ga yana can. Okay, so all right. So citric acid now it can either enter the citric acid cycle or the TCA cycle or undergo conversion to ketone bodies. Ketone bodies include acetone, acetoacetate, or dihydroxy butyrate. Okay. For export to other tissues, the term bodies is a historical artifact. The term is occasionally applied to insoluble particles, but these compounds are soluble in blood and urine. Okay, so ketone bodies formed in the liver are exported to other organs as fuel. Like I said earlier, And when you have low glucose, the brain uses glucose exclusively. But if in the absence of glucose, the brain uses ketone bodies, just like explained in your lecture note. So this is the pathway. This summarizes everything, to be honest. Okay. This acetyl-CoA. Acetyl-CoA can either go into 2-citric acid cycle or can be converted to ketone bodies. This is the pathway. 2-acetyl-CoA in the presence of thiolase will be converted to acetyl-CoA, and then acetyl-CoA will now be condenses with another acetyl-CoA to form beta-hydroxy beta-methyl butyryl-CoA in the presence of all these enzymes. Now one molecule of acetyl-CoA is now removed and you have acetoacetate. This is a ketone body. Acetoacetate can be converted to acetone by this enzyme, a decarboxylase, which will remove a carboxyl group, right? All right. Removing this carboxyl group here converts acetoacetate to acetone, while oxidizing acetoacetate in the presence of beta hydroxybutyrate, reducing acetoacetate, right? In the presence of beta hydroxy, D-beta hydroxybutyrate dehydrogenase converts acetoacetate to D-beta hydroxybutyrate. This is a reduction because the reduced form of this coenzyme is oxidized here. So this is reduced, right? And the enzyme that does this is a dehydrogenase. So basically, this is proof of what we are saying. Okay, so all right. Basically, beta hydroxybutyrate can be used as fuel, like we mentioned in your lecture note. Beta hydroxybutyrate is synthesized in the liver, passes into the blood, and thus to other tissues where it is converted in 3 steps to acetyl-CoA. This first oxidized to acetoacetate, just like this was acetoacetate was reduced to beta hydroxybutyrate. So of course it can be oxidized back, right? It makes sense. It is first oxidized to acetoacetate, which is activated with coenzyme A, donated to succinyl-CoA, then split by thiolase as the acetyl-CoA transformed is used for energy generation. I think it makes sense. This is very clear. This is just the reverse of what just happened here, basically. Reverse of what just happened here. That's beta hydroxybutyrate is now oxidized to acetoacetate, and acetoacetate transferase with the transfer of CoA from succinyl-CoA. You now have acetoacetyl-CoA. The function of what's going on here, succinyl-CoA will donate this CoA to acetoacetate, and that's why you have acetoacetyl-CoA, right? It has lost its CoA and now it's just succinate, right? It's a transferase. It's very beautiful, right? And then acetoacetyl-CoA is now in the presence of thiolase enzyme, it's now converted to 2 molecules of acetyl-CoA. Acetyl-CoA will go into the TCA cycle, citric acid cycle, and electron transport chain to provide energy. That's when there's a need for energy in the cell. Okay. All right. Okay, so this is just another example. Okay, lipid droplets, hepatocyte, acetoacetate, your ketone bodies, acetoacetate, beta acetoacetate, and this. So acetoacetate and D-beta hydroxybutyrate are spotted as energy source for heart, skeletal muscle, kidney, and brain, right? Just like I've mentioned the brain. Glucose exported as fuel for brain and other tissues, of course. Okay, fatty acid, acetyl-CoA, beta oxidation, oxaloacetate, gluconeogenesis. Now this ketone body formation and export from the liver, conditions that promote gluconeogenesis, untreated diabetes, severely reduced food intake, slow the citric acid cycle by drawing off oxaloacetate, and enhance the conversion of acetyl-CoA to acetoacetate-CoA. So this is mostly common in diabetic instances, right? The release coenzyme A shows continued oxidation of fatty acids. So even in the presence of diabetes, that's why when some diabetic patients breathe, they have this, the smell, have this acetone consistency because they produce a lot of ketone bodies. And probably their brain, in the absence of maybe glucose, will have to use ketone bodies, right? Uh, so I think it makes sense what they're saying here. All right. Now, this is what I've presented in the slide. I don't think if you juxtapose what I presented with what you have here, you have a better understanding of what we are talking about. Now, some of the clinical aspects of carnitine deficiency, this occurs particularly in newborn and preterm infants due to inadequate biosynthesis of or renal leakage. That's carnitine, right? The symptoms of carnitine deficiency include hypoglycemia, lipid accumulation, muscular weakness. Treatment is by oral supplementation with carnitine, basically. And what does carnitine do? You can answer that, right? Refsum's disease, it's an autosomal recessive neurologic disorder. You know what an autosomal recessive disorder is, right? Autosomes. What are autosomes? You should know, nursing students. Autosomes and sex chromosomes, right? Disorder of lipid metabolism, which occurs due to metabolic defect that result in accumulation of phytanic acid. You remember phytanic acid and phytol? If dietary lipids derived from chlorophyll. So it means if alpha oxidation is defective, you're going to end up with Refsum's disease because alpha oxidation is what gets rid of phytanic acid, converts phytanic acid to a more suitable substrate for beta oxidation. And in the absence of that, then it means we are going to have problems such as this disease, right? Refsum's disease. Affected individuals may display retinitis pigmentosa, diminished deep tendon reflexes, and incoordination. Then you have your Zellweger's syndrome. This occurs in individuals with a rare inherited absence of peroxisomes in all tissues. Okay? So you don't have peroxisomes. That means you have problems, right? And that's the site for which oxidation? You should tell me. Yeah, the accumulates C26 to C38 polyenoic acids in brain tissue. Wow, that's a very long chain fatty acid. And also exhibit a generalized loss of peroxisomal functions. This causes severe neurological symptoms, and most patients die in the first year of life. Wow. Okay. All right. Jamaican vomiting sickness. Okay. This is caused by eating the unripe fruit of ackee tree, which contains the toxin hypoglycine. This activates medium and short chain acyl-CoE dehydrogenase, inhibiting beta oxidation. Okay, this is quite interesting, isn't it? And causing hypoglycemia is caused by a lack of mitochondrial medium chain acyl-CoE dehydrogenase. These are places where we can ask you a lot of questions. I hope you are taking note. Ketoacidosis, higher than normal quantity of ketone bodies present in blood or urine, constitute ketonemia and ketonuria, respectively. Of course, ketone bodies. The overall condition is called ketosis. It is pathologic. It is pathologic in diabetes mellitus. Non-pathologic forms of ketosis are found under conditions of high fat feeding, after severe exercise and post-absorptive phase. I think this is self-explanatory. Define ketonemia as higher than normal quantities of ketone bodies in the blood. What of ketonuria? Tell me what ketonuria is in the comment section, please. I need to know. That will prove to me that you've listened to this lecture. All right. And you can read up the conclusion. If you have any questions, please don't hesitate to post in the group. Thank you. And then we'll move to study session 2 in the next meeting. All right.